Unlocking the secrets of immunity, how scientists are combining RNA and stem cells to fight viruses. So the article I chose was called Stem Cells Can Use the Same Method as Plants and Insects to Protect Against Viruses. I chose this article because due to the COVID-19 pandemic, I have developed an interest in the way viruses work and the newer tools that researchers are using to fight them. There have been some exciting advancements in the fields of virology and immunology over the last decade, and independent teams all over the world have been working with stem cells and dicer to fight RNA viruses and to teach the body how to fight them. Many of these studies include the use of animal stem cells, including organoids made from mouse stem cells and various forms of RNA. So I'm going to go over some important terms that you're going to need to know to follow along. First, RNA. RNA, or ribonucleic acid, is a single helix-shaped molecule that aids in protein synthesis. Among its many functions is carrying the genetic information for viruses. There are many forms of RNA, some of which I'm going to talk about today are including messenger RNA, or mRNA, which delivers the information that is used to build a peptide chain, which is the image in the bottom right corner. MicroRNA, or mRNA, which plays a part in gene regulation, and virus-derived small interfering RNAs, or VSI RNAs, whose role is to restrict viral infection through RNA interference. You're also going to need to know what dicer is. Dicer is an endoribonuclease protein that is encoded by the dicer1 gene. An isoform of dicer, AVID, is an antiviral dicer, which protects the tissue stem cells from RNA viruses like the Zika virus or SARS-CoV-2. It works by cutting up or dicing viral RNA, and this prevents the RNA from replicating. Some more terms are organoids and stem cells. Stem cells provide new cells for the body and replace specialized cells if needed when cells are damaged. Because of their unique ability to grow into needed cells, stem cells are used in regenerative medicine. Organoids, on the other hand, are miniature simplified tissue cultures that are made from stem cells, and they're made to replicate organs. Organoids are used in experiments to study how organs react to different stimuli, including diseases and drugs. So the original study that the article from Science Daily was talking about is entitled "An Isoform of Dicer Protects Mammalian Stem Cells Against Multiple RNA Viruses." Scientists at the Francis Crick Institute have been experimenting with new ways to fight RNA viruses. They found that mouse stem cells contain the instructions for creating the dicer enzyme, which can be used to stop replication of RNA viruses. They created brain organoids from a mouse's genetic materials and exposed the organoids to viruses in order to study the reactions. They found that the organoids were equipped with dicer had fewer infections, and in those infected organoids, the virus created fewer viral materials. The hope of these researchers is that they'll be able to use this process to teach human cells how to create the dicer enzyme and help our bodies learn to protect themselves against RNA viruses. Some other studies that are going on right now that I'm going to talk to you about is this one study that's being done right now in Japan by researchers of the Japanese Society of Regenerative Medicine. The article is entitled miRNA, or microRNA, Signaling Networks in Cancer Stem Cells. A team of researchers in Japan are using a similar strategy, working with microRNA, or mRNA, to access cancer stem cells. Some mRNAs act as natural oncogenics, or tumor suppressors, in various cancers. Using the same strategy, this time with cancer stem cells, they can actually block the oncogenic mRNAs and suppress the tumors. The biggest problem that they're finding is that the mRNAs can degrade in RNA serum. They've been experimenting with using chemically modified nucleic acids to stabilize the mRNAs and establishing a better delivery system. If successful, this can be a huge breakthrough in cancer therapy. Another study I'm going to talk about is being done by researchers from the National Natural Science Foundation of China. And this research team in China have been studying immune response to a group of RNA viruses, including those that cause Zika and influenza in mammals. Dicer is being used to process the viruses, which is being facilitated by the RNA interface. What the team is doing is they're using the production of virus-derived small interfering RNAs, the VSI RNAs we spoke about earlier, to restrict viral infection. The biggest challenge for this team is that so little is known right now about dicer processing in the VSI RNAs in somatic cells. So they need to gather more research in that area before they could advance the study. Just a few things to know going forward. The fields of immunology, virology, and microbiology have been transformed over the last decade as we've learned so much more about the types of RNA and their functions. This brings forth a new world of RNA and what it can do. One of the biggest challenges ahead of us is, as the researchers from the National Natural Science Foundation of China mentioned, our lack of data on the functions and processes of the newly discovered RNA forms, which is slowing down the research. So we still have a lot to learn and are just cracking the surface of what we can do combining dicer and stem cells. But these breakthroughs are key to teaching the human body to defend itself from future pandemics. And the future. While researching this project, I learned so much about the role of stem cells and enzymes like dicer, and I also have a much stronger understanding about how a virus replicates and the role RNA plays in mitigating the viruses. I look forward to seeing what comes of the 3 studies I wrote about and some others I discovered while doing this project, like the team that's close to synthesizing a vaccine for the HIV virus, or the numerous cancer studies taking place worldwide, and a few other teams that are working with dicer and stem cells, much like the researchers from the Francis Crick Institute. And it's just a very exciting time in the world of microbiology with new discoveries that will one day change the way we fight and prevent viruses. There are quite a few studies in different parts of the world right now that sound very promising, but one thing is for sure, the work that these scientists are doing today will improve the quality of life for so many human beings over time. And that concludes the presentation.